Rebeck from Georgetown University. Dr. Rebeck is professor of the Department of Neuroscience, co-director of the NIH jointly sponsored institutional predoctoral training program, the T32, at Georgetown. Dr. Rebeck grew up in Cincinnati, so he's from the Midwest, and attended Cornell for his bachelor's degree. And after graduating, he worked for 3 years as a chemist in an agricultural products company, and then earned his PhD in toxicology at Harvard in 1990. He spent 1 year on a Fulbright in Germany, pursuing research on Alzheimer's disease in the lab of Konrad V. How you pronounce his last name?
Bayreuther.
Bayreuther. And he then began research at Mass General with Brad Hyman, and in 2003 he moved to Georgetown, where he has been since. And for the last 25 years, Dr. Rebeck has been interested in genetic risk for Alzheimer's disease, specifically variants of the APOE gene. And his lab uses cell culture and mouse models to examine the effects of APOE isoforms on normal brain function prior to the accumulation of Alzheimer's lesions. He has been very involved in graduate education, serving as assistant director for the master's program in integrative neuroscience. He's the director of the PhD program at neuroscience for the last 8 years. And recently he's been Dean of Graduate School of Arts and Sciences. And in 2017, Dr. Rebeck received the Georgetown Presidential Distinguished Scholar Teacher Award. And in addition to talking about this wonderful science that Dr. Rebeck does, he's also going to talk to you about the different training opportunities that Georgetown has to offer to our students. So thank you so much for being here, Dr. Rebeck, Bill, and I leave it for you to present. Thank you.
So let me do what you taught me how to do.
Let's see. Good.
Okay, so
now you have to.
Now I have to do my screen.
Yes. Thank you.
Okay, so thank you. Thank you for the invitation. I don't actually get invited to give a lot of talks, but this is exactly the kind of talk that I like to give. So, you know, I hope you appreciate it, and please interrupt, ask questions. I mean, my whole point here is to be understandable. So please help me do that. Before I get started, I just want to thank my lab. So on the left here are the people who are, well, mostly the people who are currently in the lab. 2 of them are master's students and they've moved on. But I'm going to particularly try to highlight work that's been done in my lab over the last number of years by PhD students, because I think that's useful for you guys to know what's possible as a graduate student. And then there are some people who I collaborate with and I get funding from the NIH. But here is my group. It's it's also one of the people in the middle here, Sarah Flowers, has moved on to have her own lab. And I really like my group. I don't know that this is always true, but I am at a moment when I really am fond of my group. So for those of you who don't know a lot about Alzheimer's disease, there's a place, the Alzheimer's Association, which is a source of a lot of important information, a lot of help for caregivers, a lot of help for researchers. And so I pulled this from their website, and there's nothing surprising here. It's just letting you know how important of a disease this is that we're studying. It's one of the facts here is that it's a leading cause of death. But of course, that's not what really worries people. What worries them is how they have to live with Alzheimer's disease, because it's so devastating to the people and to their loved ones. And also, it turns out to be the most expensive disease in the US, which of course is a reason to spend more money on it. Okay, so what is Alzheimer's disease? So if anybody ever asks you, do you know anything about Alzheimer's disease? You can say, well, I know plaques and tangles. Like that half sentence there, no, I guess it's a whole sentence, is a pretty good sentence. So if you look at a brain of somebody who's had Alzheimer's disease or who has Alzheimer's disease, there are these plaques, which are made up of one protein that becomes insoluble outside of cells. That protein's called A beta. We sometimes call those things amyloid. But you always see amyloid, or you see these plaques in an Alzheimer brain. You also always see these tangles, which are intracellular, and the black things that you're seeing here. You could see that some of them look the shape of neurons, because they do. They accumulate inside of neurons, and eventually that neuron dies, and the tangle may be extracellular at that point. But absolutely, plaques and tangles, this is what defines the disease. My interest in the genetics of Alzheimer's disease goes back to the early 90s, and particularly a discovery that was made by Alan Roses at Duke University. And what they found was that there was this gene, APOE, that has genetic variants, and they were studying familial forms of Alzheimer's disease, and they found one of these forms present a lot in the Alzheimer patients and not much in the non-Alzheimer's patients. And we'll go through that in a second. And I was at Mass General at the time, and we pursued that research. And this was a paper that I published back then about looking at sporadic Alzheimer's disease. So we simply had whoever walked into the clinic at Mass General to be treated for Alzheimer's disease, and we looked at what APOE they had. And again, we'll get into this. There's APOE2 and APOE3 and APOE4, and we saw that a lot of the Alzheimer patients had APOE4, and many fewer of the controls had APOE4. And this was really on what, in retrospect, was a very small number of people, in the 30s in the controls and in the 30s in the Alzheimer's disease group. Since then, the field of Alzheimer's disease it's a very collaborative field. And so now people are studying tens of thousands of people with Alzheimer's disease and tens of thousands of people who are controls, aged controls. And they've identified all sorts of genetic risk factors. So the way this works is, here's a bunch of genes that have been identified on this slide, and they all have different variants. So there's just different common forms of them. And inheritance of one of these common forms will increase the risk of Alzheimer's disease, and maybe another common form will decrease the risk of Alzheimer's disease. But you can see that there's lots and lots and lots. And APOE is over here with a whole bunch of dots next to it. And the reason that I've continued to study APOE, even as people have discovered more and more variants that change your risk, is that APOE has a unique feature that makes it the strongest genetic risk factor for Alzheimer's disease. Okay, that's kind of a big statement, the strongest genetic risk factor for Alzheimer's disease. Okay, so across the x-axis here, we have the frequency of these variants. Okay, so let's take a case of a mutation in a gene. If you inherit a mutation in a gene called APP or in a gene called presenilin, and that mutation is associated with familial Alzheimer's disease. If you get that mutation, you will get Alzheimer's disease. There's not very many people in the world who have those mutations, hundreds probably, but those that do will get Alzheimer's disease. On the other hand, there are these variants that are quite common in the population. So, I mean, we all have the same genes, but those genes don't have exactly the same sequence. And when there are common sequence variations between us, we just call them different alleles, different variants. And APOE is one of those genes. So it has an allele APOE2, an allele APOE3, and an allele APOE4. Now, the variants in a whole bunch of genes change, like that I showed you in that one big plot before. Those changes to your risk of Alzheimer's disease are a few percent. So if you inherit one variant, your risk might go up by 5%. You inherit a variation in another gene, your risk might go down by 2%. If you inherit APOE4, though, your risk goes up about 3 times. So it goes up to 300%. Actually, if you inherit 2 copies of APOE4, your risk goes up about 15 times. So quite dramatic effects, and we'll come back to look at those. And your APOE4 is quite common. So 25% of us in the United States have inherited APOE4. So you could know nothing else about your genetics, but if you knew your APOE genotype, you would have a pretty good sense of how much risk of Alzheimer's disease you have. Okay, this is what this now looks like on a population basis. So this is another slightly odd plot, but beautiful. So what this is a survival plot. On the y-axis here, we have those individuals, the percentage of people who don't have Alzheimer's disease in these different groups. So at the age of 58, nobody essentially has Alzheimer's disease. But as we get older and older and older, more and more people start falling into the Alzheimer's disease group and away from the non-Alzheimer's disease group. So if you have inherited the most common form of APOE, which is APOE3, if you've inherited 2 copies of APOE3, and that's about 2/3 of the US population, that's this black curve here in the middle. And if you live to mid-80s, your risk of Alzheimer's disease is about 50-50. Okay, that is kind of a scary fact because I think all of us imagine living well into old age, and if you want to live into your mid-80s, you are dealing very likely with a chance of Alzheimer's disease. Now, if you've inherited 1 copy of APOE4, and like I said, that's almost 1/4 of the population, that will be these 2 curves here, these 2 that are coming down here. So, and the way that looks is if we stick with this 50% number, now instead of getting Alzheimer's disease in your early 80s, you're getting Alzheimer's disease, it's a 50-50 shot in your mid-70s. And if you're one of the 1 or 2% of the US that's unlucky enough to inherit 2 copies of APOE4, your risk goes forward by another 5 years or so. So these are very strong effects. Oh, and I should point out that there's also the APOE2 allele. And if you inherit the APOE2 allele, your risk decreases. And so you now have another 5 or 10 years reduced risk of Alzheimer's disease. And a few people have inherited 2 copies of APOE2, and their risk is even lower. So this is a very strong factor, genetic risk factor. And so rather than trying to study something that has a small effect, we have continued to study something that has a big effect on your risk. Another scary thing about Alzheimer's disease is that your brain starts to accumulate the lesions of Alzheimer's disease probably about 15 years before there are clinical symptoms. So let's take me as an example. I'm 59 years old. I don't know what my APOE genotype is, but let's imagine I have one APOE4 allele, which put my risks at 50-50 in my mid-70s. So then going back 15 years, I'm just about at the age when the amyloid might start accumulating in my brain. And so here's amyloid accumulation in individuals who have 2 copies of APOE4, or people who have 1 copy of APOE4, people who have APOE3, or and this was a meta-analysis of some very different types of studies. But again, it shows that inheritance of APOE4 increases your risk of Alzheimer's disease and increases your risk to earlier times. So my lab has taken the sort of simple proposition that, well, it's not like APOE's been sitting around doing nothing your whole life until you get to be, 59 years old, and then maybe things start to go wrong. Of course not. APOE's been doing something your whole life, and then in the presence of aging and Alzheimer's disease pathology accumulating, those functions don't help if you're APOE4. So the way I think about this is just an example of a cartoon somebody in my lab drew. And they said, okay, so here's APOE. It's part of these lipid particles. We'll talk more about that. That's why there's this little yellow circle for carrying around cholesterol, phospholipids. And one of its functions is to get rid of stuff, lipid stuff, hydrophobic stuff. And so it carries that stuff into the blood vessel and helps clear it out of your system. Or maybe it takes a little bit and gives it to glia, and the glia munch it up and get rid of it. Another thing that APOE lipoproteins do is they can act on these glia to limit inflammation. So if there's ever damage and the glia become inflamed, APOE lipoproteins are known to put a little bit of a brake on that. And then finally, APOE also, if you add it to neurons, these APOE lipoproteins, they're providing the neurons the lipids that they need to make more connections, more spines, and so they're trophic in that function. All of these functions are applicable to Alzheimer's disease. So we have this amyloid that has started to accumulate late in life for whatever reason. And APOE is important for clearing that either into the bloodstream or microglia or astrocytes clearing it. There's a lot of inflammation in an Alzheimer's disease brain around these lesions like amyloid plaques, and APOE can act to dampen that inflammation. And then APOE can be providing support for neurons that are dying, and if it doesn't do a good job at any of these things, then amyloid or then Alzheimer's disease accumulates. We are not clearing the amyloid, we are not preventing the inflammation, which can be damaging over time. We are not providing trophic support to neurons. So our starting point has been to say, let's just study these things in a normal brain, because it's a lot simpler to do that. And a lot of people are studying amyloid and tangle formation, and they don't need me to study that as well. I can study other things. So what we study are APOE knock-in mice. So these are mice where the mouse APOE was taken out and was replaced with human APOE. So they are perfectly normal mice. They express APOE perfectly normally throughout their lives. It's just they express APOE, human APOE2 or human APOE3 or human APOE4. And here's what those proteins look like. APOE is 299 amino acids long. It is secreted from cells. It's got a domain that binds to lipids, so it's part of these lipoprotein complexes. This is how your body moves lipids from one place to another, because they're lipids. If they didn't get packaged a certain way, they would just stick to the side of your blood vessels. So they get packaged into these lipoproteins, and APOE is one of the proteins that's on those lipoproteins to stabilize it and traffic it. Those differences between APOE 2, 3, and 4 occur in this domain, and they're just single base pair changes that lead to single amino acid changes. So there's either cysteines at these positions or arginines at these positions. So the proteins are not very different. The mice are not very different. And when they express APOE, it gets expressed all over the place. So here's just a Western blot we ran looking at different tissues from these mice. And this is cerebral cortex, but then a lot of peripheral tissues as well. There's a lot of APOE in the plasma for these lipoproteins. This is just TBS, meaning there's no detergent when we grind up the cerebral cortex, which means we're looking at APOE that's very soluble. And then if we add this X, Triton X, that's a detergent. And so this APOE is more like APOE that's stuck on membranes somehow. Lots of APOE, lots of tissue. But in the brain, it's mostly expressed by glia. And so we took primary glia from these animals, and we looked either at astrocytes, and that's the top here, or we looked at microglia, and that's the bottom here. And we grew them in culture, and the glia either came from APOE2 mice, or APOE3 mice, or APOE4 mice. And we just measured how much APOE gets secreted by these different cells. So the astrocytes, actually, if you look at the axes here, the astrocytes have got much higher numbers than the microglia. So astrocytes secrete a lot more APOE than microglia do. And then the APOE2 microglia secrete more APOE than the APOE3 microglia or the APOE4, or the APOE2 astrocytes more than the APOE3 astrocytes, more than the APOE4 astrocytes. Looking across both astrocytes and microglia, you see the same thing. So there's less APOE4. And this is work by a postdoc, Maria Frey Lanfranco, but also a graduate student, Jordi Sepulveda, who joined the lab this year. The APOE that gets secreted is also associated, the APOE4 is associated with smaller lipoproteins. So this was a study where we took cerebrospinal fluid and looked at the size of the lipoproteins that APOE was on. And the way that this is defined is this is how much APOE we saw, and this is sort of how big the lipoprotein was. So this is like a really big lipoprotein over here, and over here would be a really small lipoprotein. And when we looked at CSF from individuals who had an APOE2, they had a distribution, some very big, some smaller, but on the whole, there were larger lipoproteins compared to the APOE3 individuals, and then again significantly larger than the individuals who carried APOE4. So not only is there less APOE4 in being secreted, but the APOE4 that is secreted is on smaller lipoproteins. Now, you may have noticed from a couple slides ago when I talked about, I'll go back just to show you that I'm not lying. When I talked about APOE in the cortex, you might have noticed, well, there's kind of this smeary part here, which honestly, I think people just have ignored for years. I know I've mostly ignored it for years. But it's generally not present in other tissues. And we were particularly started to pay attention to it because a student in my lab, Amanda DiBattista, looked at APOE that was taken from cortex of APOE3 mice or APOE4 mice. And she noticed that the soluble APOE was always bigger than the membrane-bound APOE. And this was true if she did it from mice brains, and then it was true if she did it taking human brain samples from individuals with different APOE genotypes. So what does that mean, that it's bigger and that it's smearier? Well, one way to find out what proteins look like is to tear them apart into tiny pieces, and you do that by mass spec. So this is when Sarah Flowers joined my lab for a while, because she was an expert in mass spec and had gotten interested in this project because we had been seeking her help and trying to figure this out. And so these are the kinds of analyses that she would run with her mass spec. And basically, she identifies fragments of APOE, and if there is something added to them, that will, of course, change the molecular weight. And in this case, sugars are added. And in this case, sugar is being added to a threonine residue. And it could be a sugar that has got these two, an oligosaccharide with two components, or with three components, one of which is charged, or with four components with two charges. So the glycosylation, the glycans that are added, can be quite complex. And this fragment here was actually one that people knew 30 years ago, that APOE was glycosylated at that site. And I think we all ignored that smear for a long time because we figured that's just what it was. But that's not what Sarah found. Sarah found that there's O-glycosylation at several places. So O-glycosylation means that you're adding a glycan either to serine or threonine in the case of APOE. And so there's O-glycosylation that happens at the N terminus. There's this O-glycosylation that happened in the middle that we knew about. And then O-glycosylation that happens in this lipid binding region all the way at the C terminus. And this is the different kinds of things that could be added at those sites. And she compared what she found in cerebral spinal fluid, and that's up here, looking at the N terminus, the middle part, or the C terminus, with what she saw in plasma. And there were a lot of differences, but let me just draw your attention to what's going on here. If we look at APOE in the plasma, in this region, this lipid binding region of APOE, almost none of it is glycosylated. Whereas if we look in plasma or in CSF, nearly half of it is glycosylated. And that, of course, is potentially very interesting. So we're adding sugar in this region that interacts with lipid. And so this is just a cartoon that she made to look at the structure of APOE. And I won't go through it all, a lot of alpha helices here. But this, the little gold balls, are what a glycan looks like, the glycans that she was identifying. And I included here to point out that it's not a small change to the protein. I don't think I had ever really paid attention to glycosylation very much, and I assumed, okay, sometimes you add a little sugar somewhere. But it's really quite large, and in this case, this one happens to be doubly negatively charged. And so while we say this domain of APOE will be interacting with lipids, adding a few negative charges there is going to completely mess up that interaction. So this is something we're continuing to investigate. What does adding negative charges here in the lipid binding domain, what does that do to APOE functions? So up to this point, I've been talking to you about APOE, and I've talked about APOE expression, so APOE levels that we see out in the outside the cell. We've talked about APOE glycosylation, adding these glycans to APOE, and then APOE forming these lipoproteins with all these lipids. And then, you know, this providing, I've implied that it provides lipids to neurons to help them grow. So is there any data about that? And so, of course, that's something we've been interested in. And in this case, we used this thing called Golgi staining, which is 100 years old and just beautiful. And it's a way of looking at some neurons in the brain and looking at everything about those neurons. So here's a neuron that's stained, and you can see dendrites, you can see branches off those dendrites, you can see dendritic spines off of those branches. And so you can really get a sense of the complexity of neurons. And a few years ago, I had a couple of students who were interested in these processes, Sonia Demanis and Gus Rodriguez, who looked at the complexity of neurons, and maybe they looked at dendritic spines along these, or they looked at how many branches or how long those dendrites were, and reproducibly found that the neurons from APOE4 mice were simpler than the neurons from APOE3 mice and simpler than neurons from APOE2 mice. And so that, again, fits with this idea that APOE4 is deficient a number of ways. It's been deficient in secretion, been deficient in lipidation. Now it's deficient in helping neurons form connections. We then moved on and said, well, so if we're seeing differences in neurons, we should be seeing differences in behavior if we're smart enough to look at the right behavior. So we looked at this Barnes maze, which maybe some of you know, but basically you put the mouse in the middle of a white table, and the mouse doesn't like to be exposed. And so it runs to the side, and this one is thinking about jumping, but it's too far down. So instead, the mouse will go around and explore these little false holes, thinking maybe one of them will let me escape. And indeed, one of them does. And it's always the same hole. So once the mouse learns from looking at these other things in the room that the hole is over here, then once you drop the mouse in the middle of the maze, it'll take a second or two to figure out which way to go, and then it'll go right to that hole. And so this is a way to measure spatial learning and memory. And so this was something that Gus did in the lab a few years ago, and put the mouse in the beginning, and on the first training days, the mice look around at a lot of different places, and you leave them there for up to 3 minutes, and if they don't find it after 3 minutes, you give them a push to where the hole is. And so the first day it takes them, they learn over a few trials that first day, they start to learn. And then they learn more by the second day and the third day. And APOE2 mice and APOE3 mice learn how to do this quite well. And APOE4 mice generally learn how to do it, but they're slower at it. And this, it always scares me when I talk about this, because I think, you know, what if I'm an APOE4 person? Maybe I'm just not good at certain learning skills because that's my genetics. And I think that is probably true. I think our genetics affects all sorts of things that we take for granted. So of course it would affect our cognition in some way. So this is just something that I worry about when I need other things to worry about. Okay, that was all genetics. That was like straight up genetics. But of course you don't just get any diseases for the most part because of genetics. Usually there's some combination of genetics and environmental risk. And so two of my current, my two current graduate students, Nadia Jones and Tamar Demby, have been looking at different risk factors for cognitive impairment. And Nadia has been interested in obesity, which is a risk factor for Alzheimer's disease. And Tamar is interested in cognitive impairment that goes along with cancer chemotherapy. And that's a new topic, so we'll come to that, but we'll start with obesity first. So the basic idea is, well, let's mix a genetic risk factor with an environmental risk factor and really try to do what we all talk about, and that is, you know, how much is something nature and how much is it nurture. So she put her mice on high-fat diets, and this is how long mice have been on a high-fat diet, and then this is how much they weigh, and blue is APOE3 and red is APOE4, and if you put a mouse on a high-fat diet, it gets fat. There's a little bit of a difference between females and males, but they get fat. She looked at glucose tolerance, which is if you expose the animals to a little bit of glucose, how quickly do they clear it from their systems? Because we all take it up to get it out of the bloodstream and store it if possible. But on a high-fat diet, glucose tolerance goes down. So this shows that she knows what she's doing, but this is nothing about the brain, of course. So then she started to look at the brain. And one of the things that we've been interested in is gliosis and brain inflammation. And so she looked using this GFAP antibody for astrocytes and said, how many astrocytes do I see in an APOE3 brain compared to an APOE4 brain? And then this is on a low-fat diet and a high-fat diet. And you can see that looking at different brain regions, these are all hippocampal brain regions, the amount of astrocytosis increases in the presence of high-fat diet in the APOE3 animals. The APOE4 animals, we're not quite sure what to make of yet. There doesn't seem to be much change, low-fat to high-fat, but I'm not sure if that's because the APOE4 baseline animals have a little bit of gliosis. And so that's something that we're looking at. And then there's basically the same pattern in microglia where being on a high-fat diet actually changes the inflammatory state of your brain. We're trying to figure out what that means in terms of function of the brain. And so we're looking for genes that maybe are modulated in the presence of high-fat diet. And Nadia's become interested in these immediate early genes. And so these are three of the most common ones: CFOS, ARC, and EGR1. And it's the same pattern that you just saw with gliosis, and that is there's a strong effect of diet on the expression of these immediate early genes in the E3 animals, and then not so much in the E4 animals. In fact, really none in the E4 animals. And we don't know much about immediate early genes. This is not where we imagined our research to be going. But the bit we've read so far is that it's really important in synaptic plasticity, which of course is, of interest in cognitive impairment. The other marriage of genetics and cognitive impairment came about because an oncology researcher at Georgetown, Jean Mandelblatt, came to see me, and she's been studying for years these cognitive problems that people who are undergoing chemotherapy for cancer experience. So a large percentage, there's not a lot of studies, so they're quite variable, but a large percentage, 15%, 70%, like I said, a lot of variability, have problems with attention, learning and memory, multitasking. They talk about being in a fog a lot, and sometimes those problems resolve when somebody is off chemotherapy, and sometimes they persist. And this is the data that Jean brought to me. And what this is are different groups of people, and she was studying their cognition at baseline, that is, before they started to receive chemotherapy or some sort of treatment for cancer, and then 12 months later or 24 months later. And then the y-axis is some measure of their cognitive function. And this is a combination of attention, processing speed, and executive function. So they did a number of different tests, and they combined them all into one score. And the control individuals who are in blue, you can see, so these are people who don't have cancer, who just agreed to be part of this study. And over 12 months or 24 months, they get better at these tests because they just practiced them. They learned how to do them the first time. They got a little bit better, then they got a little bit better. Somebody who was being treated with hormone therapy for breast cancer acted just like controls. So they got better. People who were treated with chemotherapy, some of them did quite fine, and some of them did terrible. And the ones who did terrible were the APOE4 individuals. And the ones who were fine were the APOE4 negative individuals. So Jean brought this to me and said, we need to study this. And another group had found this as well a few years ago, but on a small study, and I think we all managed to ignore that study. But we couldn't ignore this. So we went back to our APOE mice, and I said, okay, so the easy thing to do is to just give our mice chemotherapy and then look at their cognition. And this was a graduate student, Andrew Spidell, who's now getting his PhD in another lab at Georgetown. So he gave Doxorubicin to these mice. Now these are mice that don't have cancer. So the oncology people are like, well, but this isn't what happens. And I kind of shrug my shoulders and say, yeah, but this is science. I'm going to change one variable at a time. And so this is the study that Andrew undertook. And then these data are actually from Tamar, because she did the study that Andrew did, but in different aged mice. And so this goes back to our Barnes maze. Here's how many days animals were running the maze, and this is how long it takes them. And really by the first day, the APOE3 animals have learned how to get out of the maze pretty well. So by day 2, they're really good at it. If you give chemotherapy to the APOE3 mice, it doesn't matter to them at all. The APOE4 mice, as we saw before, are worse at this task. So they don't do very well on day 1. By day 3, they're doing pretty well, and by day 4, they're fine. But if you give those mice chemotherapy, they are terrible at this task. And like I said, we've done this in 6-month-old mice, and Tamar did it in 1-year-old mice. There was basically the same finding. So we thought maybe age would make things worse, but it didn't. But really, in both cases, the combination of chemotherapy and this genetic risk factor really strongly affect the behavior of these mice. So exactly what had been implied from the human data. So just important in that sense of here was two completely different approaches to asking the same question, and they converge, which gives you a lot of confidence that what you're seeing is real. What Tamar's been doing lately is trying to look at the mechanisms behind these effects. And so she's taken a large number of animals, treated them with chemotherapy, and then at 1 day or 3 days or 7 days or 21 days after exposure, she's collected blood, she's collected brain, and she's trying to identify what is changing. What are the things that might be responsible for these strong behavioral effects? We're also doing experiments in mice that actually get amyloid. Everything I've been talking about up till now are mice that don't get amyloid. They're not Alzheimer's disease. But we decided we needed to look at amyloid mice as well. And that's just, that's been COVID has hurt us there a little bit. So right now, my students are trying to find these changes. So we've done sort of a non-directed approach to looking at neuroinflammation. Whole bunches of genes were analyzed, and now we're going through them in small groups and saying, which do we find that are the most interesting? And so far, we've hit on a couple genes involved with senescence and a couple of these genes involved with these early immediate genes, and so immediate early genes. And so we've got hypotheses to generate based on these. And then, of course, follow up on these mRNA findings with studies of proteins. So this just brings me to my last point here, which is something I've been interested in for years and years, but haven't known how to move forward. And that is, how do we take somebody's genetic predisposition for Alzheimer's disease and use it to help them prevent Alzheimer's disease? If you think about it, it's kind of an impossible experiment to do. You can't find somebody who's APOE4 positive and ask them to take an experimental drug for years and years and years and follow them until maybe they get Alzheimer's disease. Nobody would do that. And because inheritance of these genetic risks is not for sure going to give you Alzheimer's disease. However, when you're talking about chemotherapy, people might do that. So imagine that you are APOE4 positive and you know that your chances are high for having a cognitive problem with chemotherapy. And somebody says, look, I could give you a prevention approach, like just for right now. And then we'll follow you for a matter of months to see if your cognition gets worse or not. And so this is an approach that a lot of people talk about as personalized medicine or precision medicine. That is, if we know something about you, we know that you're APOE4 positive, maybe you should be taking a drug that other people wouldn't take. And I think if we can move this forward in the chemotherapy world, then we could think about moving this forward in an Alzheimer's world, where the effects take so much longer to materialize. Okay, so that's my talk. I'm going to stop sharing for right now so that we can have a little break, but I will share later when we get to the other part.
Great.
Isn't that smart?
Yes, very. Thank you. Thank you, Bill, for that very interesting presentation, because it just brings to light many aspects of how to understand Alzheimer's, both from the standpoint of, you know, what we have as everyone without the condition, so how we can be susceptible. And then it's in particular the part that made my curiosity even spark even more are both the fact of the APOE4 makes you harder for you to learn if you have intuitive with you. You know, it's not like you have Alzheimer's, it's just that you have this genotype background, and so perhaps that it impacts, you know, the way you learn and the way you remember things, and most certainly.
Yeah, it's a world. I think the students here are going to. So I don't know if anybody has, you know, taken their spit and sent it off to 23andMe or whoever does it these days, but they will tell you your APOE genotype. They might not tell you in that many words, but you can look at the data and find out your APOE genotype. And so I'm imagining thousands, hundreds of thousands of people, millions of people who will know their APOE genotype their whole lives. And so what that's actually doing, like your whole life, I think we will slowly start learning this about many genes, but I think APOE is going to be at the forefront of this.
And you think that APOE4 is a candidate for CRISPR in the future?
Yeah. I am always afraid of things where people are changing DNA. One of the things that people have been investigating, which I find interesting, is maybe introducing some APOE2 into the brain. So putting in some virus that will express APOE2 in the brain, so not changing your germline or anything, but so adding something to each individual. I think that's a smart approach. But yeah, I get afraid of DNA. I'm afraid of DNA.
Yeah, but it seems to be the next step, the giant leap of understanding, you know, biological function, it seems. There's a question, it says, we can relate APOE positive to lower cognitive ability then?
Yeah, well, so my approach to the brain is, our brains are amazing. I know we all get like sort of caught up in, oh, that person is good at math compared to that person or something. I'm like, what your brain accomplishes every moment of the day is so incredible that if one thing is a little bit more incredible than something else, it's kind of irrelevant. So, and you know, there's so many different things that the brain does that I think, all right, so maybe spatial learning on a Barnes maze is a little bit impaired in one thing. I don't know what that looks like for human beings. And I don't know what it looks like across aging. There's this interesting hypothesis that's out there that that some of these genetic variants are useful early in life, which is why they're around in high proportions. But then as you get older, they harm you. You know, these are interesting hypotheses. I don't know how to test them, so I tend not to think about them so much. But honestly, I feel like differences among DNA, like there's going to be advantages and disadvantages. And so, you know, like just look at everybody and say, wow, what we accomplish all the time is amazing.
Mm-hmm. And so there's another question. Do you have any preliminary data about how glycosylation of APOE might affect its function?
Yeah, that's a great question. So we did those analyses that that I showed you on human CSF samples that we got from Washington University in St. Louis. So that was, or no, yeah, we did that on a couple of samples we got from them. We wanted to look at function, and so we thought, okay, we don't get a whole lot from CSF, but why don't we grow cells and collect APOE as they're being secreted by glia, because, you know, that's where they're going to be glycosylated. And if you look at APOE that's secreted by glia, there is, you know, about the right amount that seems to be glycosylated. My postdoc at the time, Sarah Flowers, convinced me that mice are not humans. And most of the time I do not listen to that. And I say, no, come on, mice, humans, we're kind of the same. But she convinced me that actually glycosylation in humans is very different than what you see in mice. And so we had to take a step back and get astrocytes from humans, which the way you do that is you do those iPSCs. So those cells that you induce pluripotent stem cells. So you take cells, you make them into stem cells, and then convert them into different kinds of cells and convert them into astrocytes. So we've been learning how to do that. We collaborated a little bit and so that we could learn how to do it. And so now that's what we're doing. We're collecting all that stuff in order to be able to do the functional assays. The functional assays are not simple. So if you're looking at how well it binds to a lipoprotein, the lipoproteins all have very different shapes, very different sizes. Amyloid deposits have different shapes and sizes. So that's what we're trying to come to grips with. I know you asked me, do we have preliminary data, but we don't even have that yet. Yeah, this is my approach to giving talks, is I kind of tell you everything that I know. I mean, there's lots of things I do know that I'm not telling you, but like old things. If there's something new going on, I'll just tell you if I believe it.
Very good. Okay, haha, that's fine. Thank you, sis.
Yeah.
She's one
You're
of
welcome,
our.
Viviana.
Yes. And I meant to ask you, in not just studies, did she measure the levels of cholesterol in the high fat diets animals? The reason why I ask is because, you know, high levels of cholesterol also in the brain can induce the production of neurosteroids that can also affect GABAergic intervention and, of course, have effects on behavior. So any thoughts on that?
So absolutely. One of the, so the brain uses about a quarter of the cholesterol in your whole body. There is so much cholesterol in your brain. The thing is, most of that cholesterol is in myelin. So all those axons that are myelinated, there's so much cholesterol in there. And I think you and I in this question don't really care about myelin. What we care about is the cholesterol that's secreted, or the cholesterol that's in cell membranes or at the presynaptic, postsynaptic terminals. And so the first thing to do is to try to get to the populations of cholesterol that we would be most interested in. I haven't done that. I'm not a good enough biochemist to feel like we could do that well. We have sent a lot of brains off to somebody who I think is a good biochemist. But, you know, again, COVID hit. So I don't know where that stands. It's a great question, you know, because, you know, I think one of the things I really believe is we are not transporting these lipids around as well as we should. And so we are not delivering them to neurons the way we should. And there must be deficits that are occurring because of that. But we won't understand what those deficits are until we know what those lipoproteins look like.
Okay. And one last question, so we go to the part of recruitment. Does the APOE4 positive tends to affect more in males or in female mice? Does it affect both equally?
So in humans, there was a big study, you know, retrospective, you know, meta-analysis of the effects in humans, and there is a strong effect of APOE4 both in males and females. It pushed females a little bit. It had a stronger effect earlier in life than in males by 5 years or something, but strong effect in both sexes. And we've never noticed an effect or a difference in our male versus female mice.
Okay, thank you. So those sums up the questions for the science part. I don't know if...
Okay, I'm going to go back to my screen then and pick up where I left off. Okay, and then I go here. So Carmen mentioned that I've been a member of this neuroscience PhD program at Georgetown for my entire time there. I directed the program for about 8 years, but had all sorts of, you know, other jobs in the program. And it's something that I love. And I think a lot of the faculty, our neuroscience faculty, just love it. I think if you wanted, I think there are places you can do research and not do teaching. And that's not where we are. And I think the reason we are where we are is that we love the teaching aspect of what we do. And so this is one of the places where we do that in our PhD program. And so if you go to our website, there's a lot of information about the program. There's about 50 faculty members. It's a broad program, so there's some of us who are very molecular, some who do a lot of animal behavior, some who study human cognition. And personally, I have just loved that because I don't know if you got this from the introduction, because I don't always pay attention to introductions, but if you did pay attention, I was not trained as a neuroscientist. I became a neuroscientist because I got interested in like the molecular biology of Alzheimer's disease. And so I've been becoming a neuroscientist for the last 25 years of my life. And I love being part of this program because people will talk to me about face recognition, or people will talk to me about epilepsy or synaptic neurotransmission. I love that about this program. And so those of you who like want a broad neuroscience background, you should look at programs that have broad neuroscience faculty. Our students finish in about 5 years. They publish well. We pay attention to making sure people publish. We have about 50 students at any given time. Each year, there's 8 to 10 students who join our classes. And I think we have a few more than 78 graduates. We've been around. We were going to celebrate 25 years at Society for Neuroscience, have ourselves a party with, because we stay in close touch with our graduates, and people were excited about being in DC, and we were going to have a big party, but that didn't happen. But enough graduates to like, well, first you just love seeing them move on in their careers, but also it makes you feel good about the program. This is what our graduates look like as they finish, and then what they do later on. So important facts here are that about 2/3 of our students do a postdoc after they finish their PhD. And then a reasonable fraction of them go into science policy as well. You can imagine being in Washington, D.C., there's opportunities as a graduate student to be involved in policy, on Capitol Hill, Society for Neuroscience, AAAS are all here. And so a lot of our students get interested in being involved in science policy. And then as you follow our students longer and longer, most of them stay in science. A very small percentage don't stay in science. But of course, we all do a wide variety of things. So, and this is the percentages. I don't, some are teaching, some are academics like me. But we certainly want our students to get the skills that they need to do what they want to do when they leave. I think that's been our attitude. These are my students. I mean, it's a little unfair to just say, oh, here's a little slice of the program. But I'm proud of my students. And one of the things that you guys should look at is, what do all of the students do? Not just some programs, people will tell you stories. I can tell you stories about, one of our recent graduates who immediately became an assistant professor at University of Pennsylvania. Or right now, I'm proud of this woman, Lauren Orefice. I was on her thesis committee, and she is the Eppendorf Scientist of the Year. She studies autism at Mass General. But I see her picture in advertisements, and I'm like, no, that's Lauren. I helped her. But you don't care about the best stories. You care about everybody. And so that's why I'm showing you those statistics. And this is why I'm showing you all of my students, because you care about what everybody does. And I'm proud of Teal, who became a freelance science writer. The same way I'm proud of Anna, who's an assistant professor in South Carolina. One thing that I think you should also look out for is whether students are getting grants. We make sure that our students apply for grants. And in general, well, my students have been successful at it, but this is a skill you need to accumulate. So you just want to make sure when you go someplace that you get trained in writing grants. This is the campus of Georgetown University. There's a Carnegie Mellon rating, and the top of the rating is what they call R1 institutions, which are very high research universities, and Georgetown is one of those. I include this picture though because we have the medical center up here, and then the undergraduate, the main campus down here. And since we're an interdisciplinary program, you know, some of our researchers are like work in the psychology department or work in the biology department or work in the medical center. We are all across these campuses here. But it also means that you get to interact with undergraduates, which I think is a good experience as a graduate student, to be training undergraduates when they come to the lab. I think it's also helpful for all of us because this close connection with a campus with a lot of undergraduates means that there's a lot of resources that go toward good teaching. And so a lot of our students take advantage of teaching opportunities. We don't have requirements for teaching, but there's a lot of training that can be gotten if that's something you're interested in. And then I like to point out that Georgetown is I don't know if I say this, is a Catholic Jesuit university, which mostly does not affect sort of what happens on a day-to-day basis. But there is a social justice history with the Jesuits for 500 years. And so it is, I think, part of our training and our opportunities for some outreach. Here we are in Washington, D.C. Georgetown is up in the corner here, but within walking distance down to the National Mall. And I moved here from Boston 18 years ago, and I loved Boston, but I really love Washington. So, and of course, there's all sorts of opportunities. It's an interesting place to spend a little bit of your life. So applications are due in a couple of months. We don't require the GRE. There is a fee, but if you ask us to waive it, we will. It's just you have to ask us. We like people to show that they are interested in our program, because it does cost a little bit of money to process. And so we don't want to just, we want you to ask. The stipend is enough to live on. Our students generally share apartments, but it's, yeah, I don't think you're ever going to like make money, save money as a graduate student. We do whatever we can to help you. As I said, the program's about 50 students, 8 or 10 per year. A number of those are MD PhDs. There's also a pharmacology PhD, which many of the faculty are mentors in both of those programs. And so it's worth looking at that program as well. Coursework for a year. Then the second year, there's a few elective courses that you take, 3 rotations, and you should definitely do rotations and take them seriously, because I've said this before, but mentors are trained to be scientists, and we're not trained to be mentors most of the time. So somebody could be a great scientist and a terrible mentor. And honestly, right now, you need good mentors. You will not do your best science as a PhD student. The best science in your life will be later on. What you want to do as a graduate student is get trained. Get trained in the skills, get trained in the thinking, get trained how to do science. And then as you move along, you will use that training. We do actually now make our mentors take mentoring training, this thing called entering mentoring. So hopefully our mentors are getting good at this. But anyway, then you get into thesis research sometime in your second year. And we have various support mechanisms that we've put into place. We have student mentors. Our students are very engaged. And part of that is because we built the program where students have they have voting input into things about our curriculum or our admissions, or the executive decision making. And so they're like, it's a very tight-knit group. And there's specifically one committee that is meant for students to go to if there's any difficulties with anything. There's a part of our university is dedicated to taking care of career development. Some of that is individual development plans that you guys will follow as you go through your graduate careers. How am I doing? What do I want to learn that I'm not learning yet? But then there's other things like, how do I write a good CV? How do I interview well? We invite some of our students back to talk about their careers. One of my students is in biotech, and she came back to talk about like what that transition looked like. And then as support, you guys, wherever you are, you should lean on your thesis committee, because you can't your mentor is not the only person who knows what good science is, but hopefully you'll always use your thesis committees. So here's a bunch of contacts. Our admissions director, Ludisha Malkova, Kathy McGuire-Zeiss is our director these days. Patrick Forcelli is the director of our PhD in pharmacology and physiology, so that's a good program too. My student, Nadia Jones, is a current IPN student, and she's happy to be contacted. And then I made sure that Kayla Singleton and Edith Brignoni would also answer questions. They're Endure students who have recently graduated from our program. Kayla is at Emory University and Edith is at Stanford. So see, I'm proud of those two. Okay, so those are my slides.
Great. Any questions from students for Bill? Anyone? You can unmute yourself. If you don't want to write it, you can unmute.
Dr. Rebeck, I was just wondering, does Georgetown offer any prep programs or postbacks?
Not structured. We've certainly, actually, what's a great thing to do is to go to the NIH. They have a wonderful postbac program, and big. And, it's one of the things that's good for us, both with students before they take their PhD and students after their PhD. So a lot of people will go up to the NIH and do postdocs. And so, people can come to the area. NIH resources are phenomenal, great training up there. And those postbacks last usually 2 years. So that's a great opportunity.
Thank you.
Mm-hmm.
Any other questions? Seem to be quite thorough on all the information, Bill.
Yeah, I'm hoping that you guys are spending a little bit of time with various websites. I have no idea what admissions anywhere is going to look like. You know, before it was, we would go through all the applications, we would invite people to campus, you got to meet other applicants, you got to meet students one-on-one, go out to dinner, do little interviews with faculty members. Like, it was a really good way to get senses of universities. You guys have a hard job in front of you because all of that is going to be different this year. So I hope you like sort of bite the bullet and take a little bit of extra time to find places that you're really interested in. And then I know we are more than happy to interact with people who are interested in our program. So if you're not sure, reach out to us, send us a CV and say, what do you think? I was looking at your website and I saw these people who I might be interested in. You know, can you tell me about your program? We love that. I'm sure other places love it too, that sense of, you know, if people are showing interest, like that's a good feeling. You're going to spend a lot of time with these people. Mentoring is hard on everybody. It's hard on you guys. It's hard on us. It's not hard. I mean, it's enjoyable, but it's labor intensive. It's time intensive. It's the best way to learn. But you want to be doing it with people who are enthusiastic. So if you guys look at our website, identify some faculty who you might be enthusiastic about, please contact us. We love that sort of attention.
Sounds like a great strategy. I encourage all to do so, because once you make contact with the institutions, the admission committee, the chair of a department, they notice that, you know, the students are interested. So that's very important. And perhaps the first step to be asked to apply, you know, please apply. I guess that's what we are aiming at. And Georgetown, of course, is a wonderful institution. It's in DC, which is one of my favorite cities. And in addition to New Orleans, of course, but DC is wonderful. And
so the other thing about that is, since the application is December 1st, you will know who our next president is at the time. So you can make a decision about whether you want to live in Washington, D.C. or not. You know, I have very fond memories of celebrating on the Mall and very fond memories of protesting my head off on the Mall. Yeah, it's an interesting place to be.
Yeah, it's a wonderful city. I enjoy it very much. And so, okay, guys. So, any other questions? No, everybody's really appreciative of your time, Bill, and having, you know, spent this time and learning about Alzheimer's and also of the programs at Georgetown, and of course telling the students that some of our MPP Endure were students at Georgetown neuroscience program. So we encourage you all to see that success. And finally, we say goodbye. Thank you, but invite you to next week when we have Dr. Susana Herculano. She's associate professor of psychology at Vanderbilt University. That she will be here next week to talk about Vanderbilt. So thank you, Bill.
Thank you, everybody. Take care.
Being here. And so I'll see you guys next week. Thank you. Bye.